So let's start with the introduction of Dr. Aparna Sudhakaran V. So welcome, ma'am. And for participants, just I give the brief introduction of Dr. Aparna Sudhakaran, ma'am. Dr. Aparna Sudhakaran V currently serve as an assistant professor of dairy microbiology at Burgess Korean Institute of Dairy and Food Technology, Kerala Veterinary and Animal Sciences University, located in Thrissur, Kerala, India. Previously, she initiated her academic career as an assistant professor in the Department of Dairy Microbiology at the Mansingh Bhai Institute of Dairy and Food Technology, Mehsana, Gujarat. With over 12 years of experience, she has taught dairy microbiology at both undergraduate and postgraduate level. Instinctively scholastic, she is recipient of numerous academic laurels, including gold medal for BTech from Kerala Agriculture University, was in divisional topper for MEMTech and PhD study as National Dairy Research Institute, Karnal. AIR first in the entrance examination for PG and PhD, notable PG and PhD. Notable fellowship including DST Inspire Fellowship Award, ICR Senior Research Fellowship Award, ICR Junior Research Fellowship, and the KU Merit Scholarship has supported her academic pursuit. As a teacher, she received the ICR-sponsored Best Teacher Award from the Institute during 2016 to 17. She has guided several MTech and MSc students in their postgraduate research. As a researcher, she is a recipient of Young Investigator Award from Probiotic Association of India 2018, Best Paper Award in article published in Indian Dairyman 2012, and 12 Award for Best Poster Oral Paper Presentation during conference organized by Professional Association. She has edited 1 book, co-edited 4 books, 10 book chapters, and published over 50 articles. She completed 2 students projects sponsored by Kerala State Council for Science, Technology and Environment. She has also served as a Secretary, India Dairy Association Kerala chapter since 2019. So this is all about the Dr. Aparna Sudhakaran V, and we are delighted to have you, ma'am. So please, your topic is bioprospective of lactic acid bacteria for enzyme production in food industry. So please, ma'am, you can continue your PPT.
Okay, thank you, Sonia ma'am. Thank you. Thank you for your introduction. So first of all, I thank the organizers for giving me this opportunity to take class in the winter school. So today I'm dealing with the topic bioprospecting of lactic acid bacteria for enzyme production in food industry. So we will be dealing what do you mean by bioprospecting? What is the role of microbial enzyme, lactic acid, LAB as a lactic acid bacteria, LAB as a source of enzymes, its application in food industry, and conclusion. So we will be covering from the basics to some basics and some recent topics we will be covering. So what do you mean by bioprospecting? So something we are exploring from the biological resources, such as microorganism, and we want to discover or utilize the beneficial compounds. Here in this talk, we will be specific to the enzyme. So if we are discovering in the some valuable applications from a natural resources, we can use the term bioprospecting. It can be for research purpose, it can be for product development or value addition, etc. So coming to the enzyme part, the microbial enzymes have wide variety of applications. As you see in the figure, it can be used in textile industry, as in detergents, and in environment for bioremediation, and agriculture for animal feed additives, and so on. So it is a very vast topic to cover the microbial enzyme in 1 hour. So I will be specific to the food and beverages and the application of microbial enzymes. So the commonly used microbial enzymes in different food sectors, like in dairy, we are mainly familiar with proteases, lactases, etc. Similarly, in other food processing or packaging, there are certain common enzymes like proteases, lipases, alpha galactosidase enzyme, like some common enzyme we use in most of the food and dairy industry. So I will be concentrating on that. Before going to that, first we will have to introduce our lactic acid bacteria. As I know you all are familiar with lactic acid bacteria, which have been co-evolved with humans. from the during the animal domestication and agricultural practices, and now it is comes under the 6th family, and recently in 2020 the nomenclature of lactic acid bacteria has been changed, and this is the gist of that. Like some recent changes, like Lactobacillus salivarius, it is Ligilactobacillus, Lactobacillus fermentum, it become Limousi-Lactobacillus. Like some changes in the nomenclature happened, and we have, when we are using abbreviation, we have to do use L, not LB. So this is the some of the recent thing happened in Lactobacillus, and more than 25 genera are coming under Lactobacillus. These are the genera coming under Lactobacillus. Even when you go to lactic acid bacteria as a whole, it is more than 60 genera. And specific when coming to Lactobacillus, which is the rod-shaped bacteria, it comes around 25 genera. So what is the importance of lactic acid bacteria? Why we are now discussing lactic acid bacteria? Due to its GRAS status. GRAS status means it is generally regarded as safe status. So we can consume it safely. And they have lot of functional attributes like probiotic attributes, and they can tolerate stress. They have simple metabolism, as well as they can metabolize a variety of carbon sources. So these properties make lactic acid bacteria a source of enzymes. And from where we can obtain lactic acid bacteria? We can obtain from our food, fermented food, plants, gastrointestinal tract, environment. From all these sources, we can isolate lactic acid bacteria, and its application in wide variety of industries going on. So coming to the main topic, lactic acid bacteria as a source of enzyme. So mainly I will be covering these 6 enzymes, two or more I will be covering more. So mainly I will be focusing amylase, protease, lipase, lactase, gabase, and phytase. There are numerous enzymes. So specifically I have selected based on the usage of it. So first one is amylolactic lactic acid bacteria. As from the name itself, we know that it hydrolyzes the amylolactic, means starch. They will hydrolyze. Starch is made up of amylose and amylopectin. So amylolactic bacteria can hydrolyze starch, and this was the first bacteria which was discovered during 1979, Lactobacillus amylophilus, and LAB has been reported with these kind of enzyme like endoamylases, exoamylases, debranching enzyme, transferase enzyme. So in the amylase and amylopectin, there is alpha-1,4 glycosidic linkages and alpha-1,6 glycosidic linkages. Depending upon the enzyme, they will cleave that bond specifically. So these starch modifying enzyme from amylolactic lactic acid bacteria belongs to mainly alpha amylases, and maltogenic amylases have been reported, and pulanase have been reported, but the most common one is alpha amylases, which I have been discussing. So starch will be converted to dextrins and oligosaccharides, and we have to liquefy the starch. Starch, usually we will go through the process liquefaction, then saccharification, then fermentation to lactic acid. This is the normal process of starch to lactic acid bacteria. Other than amylolactic lactic acid bacteria, we use normal lab. So if we are using amylolactic lactic acid bacteria, we can convert starch directly to lactic acid. So that is, we can avoid this multi-step, and in a single step, we can form starch to lactic acid. That is the significance of amylolactic lactic acid bacteria. So this is the thing I have told, that amylolactic lactic acid bacteria can direct or single step fermentation. So where we apply this is, we can direct conversion of starch to lactic acid. We can unify this step. And similarly, we can use this amylolactic acid bacteria in the fermentation of cereals to increase its durability, consistency, taste, and aroma. So in food industry, mainly in the dough preparation for bread, this amylolactic lactic acid bacteria or the amylase have been used to increase the viscosity of the dough, fermentation rate, texture and volume of the bread can be improved, its crust color, toasting quality, these all have been improved by using the amylase enzyme. And it has been used in the cakes, baking, starch syrup, fruit juices, etc. And it is used as an anti-staling agent. Anti-staling means it is becoming dry. When we keep the bread dry for so long, it becomes dry. So if it improves the softness by retention of the moisture in the bread. So it can be done by the maltose and dextrins. So when starch is converted to simple sugars by the amylase enzyme, it acts as humectants and it can hold the moisture, so thereby increasing the softness of the bread. Similarly, it is used in fruit juices as well as in beer for the clarification, to hydrolyze the starch into soluble sugars so that it appears clear. So, and moreover, in animal feed treatment also, it is used to increase the fiber digestibility. So these are some of the applications of alpha amylase enzyme. Coming to the next enzyme, proteolytic lactic acid bacteria. So from the name itself, we can understand that we have the lactic acid bacteria will hydrolyze proteins to peptides to finally amino acids. So there will be an extracellular protease enzyme, there will be a transport mechanism, and the oligopeptides will come into the cytoplasm, where intracellular protease will act and simple amino acids will be produced. And most 65% of the global enzyme market is by protease enzyme. So where in the case of lactic acid bacteria, we have this, the basically three group, that is cell-enveloped proteinases, which is outside the cell, which will hydrolyze the proteins to oligopeptide. Then there is a transport system. Then intracellular peptidases, which will hydrolyze the peptides to amino acids. So this will improve the sensory quality, enhanced digestibility. It will decrease the allergenicity, and it can liberate bioactive peptides. So these are the proteases reported in lactic acid bacteria. Both exopeptidases and endopeptidases are reported. So if you are telling aminopeptidase, it will cleave at the amino end. If it is dipeptide peptidase, it will cleave. Like similarly, tripeptide, like dipeptidases, these enzymes are present. The classification is given here. And coming to the milk protein, the application of proteases is mainly in the cheese industry, where we hydrolyze the milk protein. Similarly, the casein will be transported. The cell envelope protein, CEP, will first degrade the casein proteins and convert it into oligopeptides. And these oligopeptides will be transferred by the transport mechanism OPP, DTP-P, and DTP-T. Oligopeptide, dipeptide, and tripeptide transport system. Finally, it will reach the cell, inside the cell, where it is metabolized into amino acids. So there is some problem with the lactic acid, is that this amino acid can also be converted into biogenic amines by decarboxylation. But there should be a balance, and this biogenic amine should be degraded. It is not healthy for us. So it is degraded in the food. And we are mainly concentrating on getting simple amino acids. Similarly, in the case of same mechanism, in the case of cereal proteins, the primary proteolysis happens outside the cell, where the gluten or the wheat protein will be broken down. Then it was transferred into the cell by the transport mechanism, dipeptidase, tripeptidase, etc. Then it will be converted to simple free amino acids, which is utilized by the cells. So where are all the applications? In dairy industry, meat industry, bakery, brewing, feed industry, food additives. In all this, there is a wide variety of applications of proteases. And now the bioactive peptides we are knowing about the health benefits of bioactive peptides. So proteases are mainly used for the generation of bioactive peptides, as well as it is used for the coagulation of cheese, and it is replacing chymosin in cheese production. And moreover, the texture and development of other fermented milk products. Coming to the additive industry, it helps to for the food concentrate, protein hydrolase, soy sauce. In this, all this we are using protease enzyme. For the development of hypoallergenic products, also we are using protease enzyme. When coming to the feed industry, this fibrous degradation of fibrous animal protein, and then the eggs or bone powder, this will be degraded by this protease enzyme. Keratinolytic peptidases will degrade the keratin. This is used for the utilization of natural waste. These proteases are exploited for the utilization of natural waste. So these are the applications. Coming to the meat industry, the proteases increases the palatability of the milk. It increases the maturing time and texture, aroma, and color. Coming to the bakery industry, it shortens the kneading time, it softens the dough, and increases the shelf life. And brewing industry, similarly to amylase, it will chill the haze beer. That hazing will be avoided. It clarifies the vote. So these are some of the applications of proteases. Coming to the third enzyme, lipolytic lactic acid bacteria. So by the name itself, it will be hydrolyzing lipids. So the lipids are triacyl glycerides. It will be hydrolyzed to glycerols and fatty acids. So the lipases have high stability. The lactic acid bacteria lipases are found to be highly stable. These are some of the bacteria reported to have lipase activity. And among the enzyme produced by lactic acid bacteria, not only lipase, esterase and phospholipases were also produced by lipolytic bacteria. So what is the basic difference between lipase, esterase, and phospholipases? The site of action. So lipase will act on water insoluble triglycerides. Usually it is long chain fatty acids, they will act. Whereas simple esters, in that case, esterase will act, and phospholipids act on phospholipids. All the three enzymes where the lactic acid bacteria are reported to produce all these three enzymes, and their major application is increase in flavor and aroma in the dairy industry, meat industry, wine, etc. And in phospholipases, it is used in the emulsification process as well as synthesis of phospholipids. So in the case of flavor development, it enhances the flavor of cheeses, and it accelerates cheese ripening. It enhances the texture and softness of the cheese. And this high Lactobacillus plantarum, it is having high esterase activity, and it will generate ethyl esters, which will give a typical fruity flavor to the camembert cheese. It has been reported like that. So in the case of wine making, the lactic acid bacteria is used for famous for its malolactic fermentation. And during malolactic fermentation, esterases from the LAB catalyzes the hydrolysis and synthesis of esters. Lipase also help facilitate the removal of fat from meat and fish products, thereby improving its quality, flavor, and maturation in cured meat production. And this is a trade name, phospholipase A1 trade name is yield max. It is from the fungi Fusarium. It is used to hydrolyze milk phospholipid. Due to the it is used during the cheese production to increase yield, this phospholipase yield. But this is from the mold Fusarium. But recently, phospholipases from lactic acid bacteria have very few researchers. So a lot of opportunities to study regarding the LAB phospholipases is there. Coming to the most common enzyme produced by lactic acid bacteria, that is the beta-galactosidase enzyme. So lactose is the disaccharide which is converted to glucose and galactose by the enzyme lactase or beta-galactosidase enzyme. So it is commonly known as lactase, and what is its importance is some of the population, 70% of our population is intolerant to lactose. Intolerant to lactose means we were not able to digest the lactose sugar, and it will reach our intestine, and since it is not digested properly, it can be utilized by the other microbes in our intestine and can cause bloating and constipation, etc. So this can be avoided by degrading lactose to glucose and galactose. And there are a lot of application to beta galactosidase in milk treatment as well as in weight treatment, and galacto-oligosaccharide. That is one of the popular prebiotic. It is now used as a biomarker in tumorigenesis and used in biosensors also. Beta-galactosidase enzyme is used in biosensors also. And now application in food industry, mainly for the production of lactose hydrolyzed milk products or lactose-free products, which is suitable for the lactose intolerant people. It is used in, it reduces the crystallization in ice creams and condensed milk, thereby improving its texture. It increases digestibility and reduces the need of additional sweeteners. And next wide application is the whey utilization. So whey can be utilized. Whey is a rich source of lactose. So the lactose will be converted to other valuable products by the usage of beta-galactosidase enzyme. Another common probiotic or thing developed from beta-galactosidase enzyme is galacto-oligosaccharide or GOS. So it is an prebiotic in our food. It is having bifidogenic activity, means it enhances the growth of bifidobacteria, which is a healthy bacteria in our gut. And it is reported to be anti-carcinogenic, serum cholesterol level reduction, and it is used as a low-calorie sweetness also. And galacto-oligosaccharides are complex mixtures of non-digestible oligosaccharides known as the prebiotic sugars. Probiotic means the healthy bacteria in our gut. They require some food, that is the prebiotics. So these prebiotic sugars, one of the common one is galacto-oligosaccharides. So what are the criteria for being a prebiotic is it should be resistant to our gastric acidity and intestinal microflora fermentation, and it should reach our large intestine, and the growth or activity by the intestinal microbacteria associated with the health and well-being, means it should support the growth of probiotic bacteria. Simply, we can say that prebiotics is the food for probiotics. So galacto-oligosaccharide is trans-galactolization occurs by the beta-galactosidase enzyme. These are some of the lactose will be converted to galacto-oligosaccharides by the trans-galactosylation by the enzyme. Don't go with the complexity of the structure. This is one of the application or commercially available enzyme of beta-galactosidase from Bifidobacterium bifidum, which uses lactose, and it will convert it into galacto-oligosaccharides. The lactose milk sugar will be converted to galacto-oligosaccharide by the enzyme beta-galactosidase produced by the Bifidobacterium bifidum. So what is the application of galacto-oligosaccharide? It is used as a prebiotic, which is the food for the healthy microorganism in our gut. So this trans-galactosylation is favored by high substrate concentration and low amount of water. If there is high amount of lactose and low amount of water, this trans-galactosylation happens. Otherwise, it will go as glucose and galactose. Instead of that, we want to make galacto-oligosaccharide. In that case, we need high concentration of lactose and a low amount of water. So this is how this enzyme is found to be producing around 107 gram per liter of galacto-oligosaccharides. Another common term now we are hearing is the psychobiotics or GABA by lactic acid, which I will explain in the next slide. So psychobiotics means any bacteria which can influence our brain through their metabolites or by their products produced by them. So there is a connection between our gut and the brain. And the bacteria or the probiotics which modulate the gut and the brain axis, then they are known as psychobiotics. Probiotic means it gives a health benefit, which when administered to us at adequate amounts will give a health benefit. Among the probiotics, if some probiotics is able to modulate our gut-brain axis, then that category is known as psychobiotics. And there are several mechanisms, out of which one mechanism is the production of neurotransmitters. So GABA is one of the neurotransmitter produced in our body, and the bacteria can produce, our lactic acid bacteria can convert the glutamine to gamma aminobutyric acid or GABA, which is a neurotransmitter. Its physiological function is neurotransmission. It will reduce our pressure, hypertension, diuretic effect, and tranquilizer effect is produced, and it is a non-protein amino acid. And now industrially, the interest in GABA is increasing due to the gut-brain axis exploration. So it has been reported to have neuroprotection, anti-hypertension activity, anti-insomnia activity, pain reduction, anti-diabetic, anti-inflammatory. In all these areas, research are going on. So our gut bacteria, including lactic acid bacteria, is able to convert glutamate to gamma aminobutyric acid, which I will show you in the next slide. This is the metabolic pathway of GABA production. So glucose will be automatically converted to pyruvate and it will enter the Krebs cycle. And as an acid stress occurs, the glutamate will enter into the cell, and instead of that, glutamate will be converted to GABA, and GABA will be expelled out of the cell in the presence of the GAD enzyme. And these are some of the bacteria reported to produce GABA metabolite, which is a neurotransmitter. That is Levi-lactobacillus, Lactobacillus delbrueckii, subspecies vulgaricus, Lendi-lactobacillus, Limosi-lactobacillus, and so on. And it is reported that Levi-lactobacillus brevis can produce a higher amount of GABA, up to 205 grams per liter. And this GABA is present in our many cereals also. And if we use this lactic acid bacteria to ferment the cereals, we can enhance the amount of GABA. So it will enhance, if we take such foods, it will enhance our mood. So this is a table showing the effect of lactic acid fermentation on GABA content in fermented dairy products. So if we fermented the fermented milk, initially it is around 10-20 milligram per liter it is present. It can be enhanced to 80-86 milligram per liter by fermentation by using lactic acid bacteria. These are some of the research works held on these aspects. Similarly, in the case of cereal foods like rice bran, wheat germ, quinoa seeds, the people are using lactic acid bacteria to enhance the GABA content. So why to enhance this GABA content, that I will show you in the next slide, because in all these products like dairy products, cereal waste product, meat, beverages and other foods, GABA enriched products are now available. The functionality of these food products are increased by enriching these foods with the GABA or lactic acid bacteria, which produce GABA. So enrichment in chocolate to provide a psychological stress reducing effect is also studied, and management of hypertension by using cheese also studied, and sort of I will show you the pictures. Like see the GABA rice, it is available, and see the price, how much it is charged for 500 gram of rice. So that much value addition is there if this kind of valuable metabolites are produced. So these are all the products available in Japanese market, in which the GABA is enriched by the lactic acid bacteria or by the enzyme. These are some of the product with brown rice with 220 milligram per kg, like that. The GABA enriched foods are now available in market, and they are claiming for the sleep improvement, anti-hypertensive activity, stress relief, etc. So in the stress world, we can think about lactic acid bacteria being a friend to us. It is naturally present in many foods, and if we ferment that food with the lactic acid bacteria, we can increase the GABA content. So there is a lot of area of scope for the researchers to increase. Usually it is by chemical synthesis or either enzymatic method this GABA is produced. We can think about how to increase the GABA content of lactic by using lactic acid bacteria. There is a lot of application regarding the use of lactic acid bacteria and the GABA enriched foods. Just to give a glimpse in these research areas. Another enzyme is phytase. So phytase or phytin is naturally occur in our fiber-rich plants, and it is considered as an anti-nutritional factor. So it will bind the minerals. So it won't be available to us. When the phytate will be bind the trace elements like trace elements as well as calcium and phosphorus to it, and it won't be available to us. So when phytase will act, this and minerals will be made available to us. So another area of research is the production of phytase by microbes, especially lactic acid bacteria. So it has been used in the bread processing, novel transgenic plants as probiotics, which is having phytase activity, grains, wheat milling, etc. So in bread processing, it is reported to have increasing its specific volume, crumb firmness. It increases the nutritive value by liberating the metals or minerals. And it is used in transgenic plants, and it is used as a probiotics. In the probiotics with the phytase activity. Similarly, it can be isolated from plant proteins. Higher plant yields, complete removal of phytase can, since it binds the protein, if phytase activity is there, it can increase the higher yield of protein from the plant proteins. It is used in plant proteins, wet meat milling. It increases the steeping processes, and the cell bran fractionation also it is used. So what is the principle of phytase is, the phytate, it is present is an anti-nutritional factor. It will bind the elements or the minerals present like iron, calcium, they will bind it. So it is not available to us, not bioavailable to us. So to increase the bioavailability, this microbial phytase will convert it, split the phytic acid to myoinositol, inorganic phosphate, and liberate the elements. So these elements will be in the food, so which will be bioavailable to us. So there are several applications of phytase enzyme, and lactic acid bacteria is also reported to have phytase activity. It can be used in two ways. We can either isolate the phytase enzyme from lactic acid bacteria, or we can, since lactic acid bacteria is having GRAS status, we can use lactic acid bacteria itself to ferment the food with the intracellular phytase enzyme. Like in the case of nuts, legumes, or pseudo cereals, we use phytate producing lactic acid bacteria, and it will increases the mineral component. So studies are going on this area. So one of the study conducted in our lab, in this we found out that the lactic acid bacteria fermented soy milk, we having phytase activity, has enhanced the iron concentration. iron concentration. These are the different isolates in our lab, and we used this lactic acid bacteria to check the to increase the mineral bioavailability of iron and zinc in fermented milk. So we were able to find out that up to 58% increase was found in one of our culture. It was able to increase the bioavailability of iron. Similarly, zinc also we were able to find out 21% increase in the bioavailability of the zinc. That is basically the phytase enzyme break the phytate, so it will hold the trace elements. So once it is broken, these trace elements or the minerals or iron or zinc which is associated with it will be liberated in our fermented product. So the bioavailability will increase. Another interesting enzyme now coming into light is the inulosucrase or fructonases enzyme. This is an enzyme. This is a study in which Lactic Bacillus plantarum is using the fructan sucrases enzyme or glucan sucrases enzyme is used, and it will convert the fructose into fructan sucrases into two types, leaven and inulin. Inulin may be familiar to you, like they are the prebiotic components in the food. So the lactobacilli is reported to have this fructan sucrases enzyme or fructosyl transferases enzyme, which convert the leaven or inulosucrase activity and produces the inulin or leaven. So these are some of the enzymes I covered. Now we will see how we can study about this enzyme. That is mainly we use omic technologies to find out the new enzymes or enzymes present in our organisms. These are the omic technologies like genomics. By using several next generation sequencing, these enzymes like protease cellulases were discovered, and similarly transcriptomics, proteomics, metabolomics using LCMS or GCMS or NMR, we are able to identify novel enzymatic processes of any bacteria or lactic acid bacteria. And what are the future things we have to do a lot? Like we have to identify novel. First of all, we have to identify novel lactic acid bacteria strains from diverse environment, and we have to study about their metagenomics and their since the yield from what is the negative of using lactic acid bacteria is its yield is very less, or it under specific condition like acid stress or some specific condition is required to produce that enzyme. So we have to enhance the expression by using molecular techniques and gene editing systems like CRISPR-Cas or synthetic biology. We can increase the expression of lactic acid bacteria. And how we will apply the industrial or food applications. There is next generation dairy processing with the increased activity of proteases, lipases, active peptides are going on. And like I have discussed, the fructonases or coarse production by lactic acid bacteria for prebiotic synthesis, and lactose-free milk or hypoallergenic products. Like it was reported that the epitope, like the proteases from lactic acid bacteria will hydrolyze the epitopes of the protein, which causes allergy. Like in wheat, one study was conducted, and in that the wheat protein, which is causing allergenicity, it's a whole protein, not the whole protein, the epitope which binds to the IgE was degraded by the lactic acid bacterial enzyme. So anti-allergenic products are now in a trend. Then it can be used for the waste valorization, that it can, like LAB enzymes can be used for converting agricultural and dairy waste to biofuels and bioplastics. And it can, okay, next is probiotic or functional enzyme. If we are having a lactic acid bacteria with the several enzyme properties, it will go into our gut, and inside the gut it can make many positive or beneficial changes. And it is also used in the bioremediation purpose. So we have to study more regarding the metabolism of lactic acid bacteria, and we have to know the ways to increase the expression or increase the enzyme production from lactic acid bacteria. So like in the microbial enzyme, it is always in trend. Because now we are thinking about clean labeled foods. We don't want any additives, or we want a natural thing. So microbial based enzymes are bio based additives. So there are a lot of advancements in biotechnology which help us to identify this microbial enzymes. These are some of the market players of microbial enzymes in the slide I am showing, and it's a promising avenue for many of us in the area of research. As you can see that enzymes are extracted based on the source from, and microorganism is the most important source for the extraction of enzymes, and its growth is increasing only. So there is a lot of avenues for us. And I conclude the talk by saying the microbial dark matter. What I mean is, like dark matter in physics, we know very little about the microorganisms around us. Less than 2% of the microbial species in our environment, we are able to cultivate it. We are able to do it in our laboratory. The rest is unknown to us. So there are a lot of enzymes to be explored. Even when we comes our human microbiome, we were able to discover only around 5,000 or 2,000 species due to the high throughput sequencing methods. But rest is hidden to us. So there are a lot of area to be explored to study in the area of enzymes, and typically from lactic acid bacteria, because from where I start, that lactic acid bacteria is having a GRAS status, generally regarded as safe. So we can use as it is in food, and it is reported to have tremendous probiotic attributes which enhances our health, and it can also produce enzyme which can produce prebiotics, which is the food for probiotics. So I conclude my topic. Thank you. Hello.
Hello, ma'am.
Hanji.
Hello, ma'am. Your slide
Yes,
is
sir.
not visible, ma'am.
Madam, I finished my topic, madam.
Okay, okay. Okay, so now it's a time for a
Any
participant to
questions?
interact with the speaker. Yeah. Any questions? One question is there, ma'am. One question is from Jala Jakshi Kuprati. Are GABA enriched product available in our market in India?
Madam, I have searched in the... Madam, GABA enriched rice is available in Amazon, and oolong tea is also available. I have searched yesterday in internet. It is available in Amazon also, but very costly.
So is there any other questions from the participant side? I think no question is there, ma'am.
Okay.
So thank you, ma'am. Thank you for your very, very informative presentation. And I think the participant must be benefited by your valuable information. And it was a very nice presentation of you. Everyone is showing his thumbs up and clap on your presentation. So congratulations, ma'am, for that also. And now you can take your leave, ma'am, and do your another important work, ma'am.
Okay, Sonia, thank you once again for giving me this opportunity. Thank you so much.
Thank
If
you, ma'am. Thank you.
anyone needs query, ID
No,
sir, they
I
can
don't
contact
think-
me.
Yes. Yeah.
Thank you.
Okay. If anyone have any query about this presentation or any other related to the field of ma'am, then you can ask on the group, and I will give you the contact number of ma'am. You can talk her directly on that problem or aspects. So thank you, ma'am. Thank you once again.
Thank you. Thank you. Thank you.
होता है आंसर बुकलेट्स फालतू का रखने का भी नहीं
Hmm.
लोग यहाँ कर लिया